Biology 9700, paper 4, variant 1, May June 2024, question number 1 and question number 2. Question number 1 is, oxygen is needed for aerobic respiration in cells, in eukaryotic cells. The mitochondrion is the organelle used for aerobic respiration. And figure 1.1 shows a transmission electron micrograph of the mitochondrion, which is labeled with A, B, C, and D. A is the outer membrane, and B is the cytoplasm, and C is the inner mitochondrial matrix membrane, and D is the matrix of mitochondria. Now we are moving to the question, and question has been shown in the table 1.1. 4 stages of respiration are given, and we have to write the letters in front of those stages. Glycolysis takes place in the cytoplasm, so it will be labeled with B, link reaction, and Krebs cycle. Both of they takes place in the matrix, so they D, and oxidative phosphorylation by C, because it takes place the inner mitochondrial matrix, inner mitochondrial membrane. The next question is aerobic respiration. Anaerobic respiration, anaerobic conditions, no ATP can be synthesized by oxidative phosphorylation because the process stops. Explain why ATP synthesis by oxidative phosphorylation stops in anaerobic conditions. Anaerobic condition, it means there is no oxygen. Aerobic condition, when there is oxygen. So we have to explain why ATP synthesis by oxidative phosphorylation stops in anaerobic conditions. You know, when there is no oxygen, so what happens? There will be no electron acceptor at the end of the chain. So when there is, the electron transfer chain stop working, and no further ATP is formed by oxidative phosphorylation. So there is therefore no free or carrier in the chain to accept hydrogen from the reduced NAD and reduced FAD. So these remain reduced. The Krebs cycle also stop working because there is no oxidized NAD and FAD to enable the dehydrogenation steps to take place. So this is the explanation. I can write it in the form of some points. So the first point can be, if there is no oxygen, so there will be no acceptor of hydrogen. Ions or electrons. At the end of the chain. So electron transfer chain will stop working. So what will happen next? So no protons will be transferred, no ATP formation, no carrier, due to no carrier acceptors in for the NAD. So NAD and AD, which are in the form of reduced form, which are in the reduced form, they will remain reduced. So Krebs cycle also stop because there is no oxidize NAD and FAD. Moving towards the next question, and next question is, up to 60% of the ATP that is produced in cancer cells comes from the lactate fermentation of glucose, even though oxygen is present. Scientists are developing cancer treatments to inhibit the enzyme that catalyzes the last step of lactate fermentation. Explain how the inhibition of this enzyme reduces the production of ATP in cancer cells. The name of that enzymes which is helpful in the formation of pyruvate into the lactate. So what will happen in this case when there is you can say in this process there is less oxygen, pyruvate act as the hydrogen acceptor. So it is converted into lactate by the enzyme which is called as lactate dehydrogenase enzyme. So we will write first of all the name of that enzyme, that is lactate dehydrogenase enzyme. So this, in the presence of these enzymes, the pyruvate is changed into lactate. So in case of enzyme inhibition, what will happen? There will be no formation of pyruvate into lactate. And if it will not be changed into lactate, so there will be no recycling of NAD, and there will be no formation of triose phosphate into pyruvate. And it is, if it is not changed into pyruvate, so there will be no ATP formation. Question number 2 is there. Coordination in the humans involve two main systems, the nervous system and the endocrine system. Paracrine cell signaling is a third way in which coordination occurs. In paracrine signaling, one cell secretes a chemical that diffuses a short distance to act upon cells that are very near to the secreting cells. Identify one similarity and one difference between paracrine cell signaling and cell signaling that occurs as part of the endocrine system. Well, first of all, I would like to discuss the nervous system takes place by the central nervous system and peripheral nervous system. And endocrine system takes place by the ductless glands, which secretes these secretions directly into the blood. And through blood, those secretions reach to the different parts of the body or to the target organs. Whereas paracrine cell signaling is a way of coordination in which the secretion move towards the other parts by the process of diffusion. So it has to cover a short distance. So the similarity which we can write over here in both systems between endocrine system, and the paracrine cell signaling is the secretion of chemicals by the system or the cell. This is the similarity. So the difference, the endocrine system the chemicals are poured into blood to cover long distance. Whereas in paracrine, short distance has to cover by the process of diffusion. So this is the difference. Explained by a neurotransmitter such as acetylcholine could be described as a paracrine signaling molecule. Acetylcholine is, it is secreted uh, from the one neuron to the other neuron, and when it is secreted from the presynaptic membrane into the synaptic cleft, then it is received by the receptor proteins, and then it enters into the post synaptic membrane. So in this way, it has to move by the process of diffusion. It has to cover a very short distance, which is only a synaptic cleft between two neurons. So we can say that it can has to move a short acetylcholine travels a short distance by the process of diffusion. So diffusion is the process by which they has to cover the, so it has to move by the presynaptic membrane to synaptic cleft, the post synaptics. So it moves from one neuron to the other by the process of diffusion. The next question, in which three experiments have been conducted, and in experiment 1, muscle cells were exposed to palmitate. Palmitate is a, you can say, molecule, and then the palmitate exposed muscle cells were removed from the medium containing palmitate and then cultured in a nutrient medium for 6 days. The palmitate exposed cells were removed from the nutrient medium and new muscle cells were placed in this used nutrient medium. The expression of stress genes in the new muscle cells were measured after 24 hours. Control using cells that had not been exposed to palmitate was also carried out. And in experiment B, experiment A was repeated, but the nutrient medium for the palmitate exposed cells and the control was blood and then cooled before adding the new muscle cells. And experiment A was repeated, but the used nutrient medium from the palmitate exposed cells and the control was treated to separate the liquid part. This liquid part was added to a new nutrient medium to culture the new muscle cells. Now we reach to the bar graph, which has been shown on x-axis. Three experimental conditions have been shown. and on the y-axis mean stress gene expression in new muscle cells. Now you can see the graph with reference to figure 2.1 outline the conclusion that can be drawn about the cell signaling molecules involved in human muscle cells paracrine signaling in response to palmitate. And you can see the condition of control was the same in all. It was on mean stress gene expression was 1 in A, B, and C. In all cases, you can say. But the medium from class exposed to palmitate was different. It was highest at B, in which it was boiled, it was at high temperature. But in the other conditions at A and C, it was at the same point, that is at 1.5. So we can conclude our results over here with reference to this picture. The expression of control medium is same in all, whereas you can see the experiment B was not denatured, although they were exposed at high temperature or they were boiled. So they show no denaturation. They were not denatured in this case. So it means they are not protein. No protein, because protein is denatured at 30 to 35 degrees centigrade. Optimum temperature is 30 to 35. At 40 degrees C, it is denatured. So we can say the 40, 35 to 35, or 30 to 35, and 40, 35 to 40. So these are the different temperatures on which the sum of the proteins are denatured, and all are denatured at 40 degrees centigrade. So in this way, we can say if this molecule which was boiled, they are not the protein molecules. So we can say they can be the other molecules such as lipids, which work best at high temperature. Now the next question. The process of protein translation is inhibited in developing muscle cells when stress genes are expressed. Suggest how developing muscle cells that express stress genes will differ in structure from normal muscle cells. You can see the process of protein translation. You can say it is inhibited developing muscle cells. When the muscle cells are not developing, so what will happen? There will be a difference between the muscle cells and the inhibited muscle cells which are not properly formed. So the muscle cells which are not properly formed, they will have the less actin. Actin, you can say this is the structure of the muscle cell in which two protein, actin and myosin are present. So there will be less actin, there will be less myosin, because the protein translation is inhibited in this case. So if the actin and myosin are less in number, so there will be less troponin and tropomyosin. No proper myofibrils or microfibrils will be formed. In simple way, the process of protein synthesis will not take place properly as it takes place the other muscle cell in which a proper uh, microfibrils or the proteins actin and myosin are present in a proper way, in a percentage, or in this way they make the different bonds, light bands or the dark bands. So in this way there is a special arrangement of these actin and myosin in the muscle fiber. But if there is no protein translation, it is inhibited, so it will be stopped, so there will be no proper formation of the muscles. So it is all about the question number 1 and 2. Please like, share and subscribe my channel. Thanks for watching.